Okay, good evening. We are going to start the next session on inflammation, immunology, and infection, oral communication. So please allow me to introduce. Okay, I'm the chair of the session. My name is Eric Chen. I'm from National University of Singapore. Our first speaker is Mohammad Farhan from the University of Tampere.
Yep. Okay. Hello, everyone. Today, I would like to talk about my presentation, and the title is TRPA1 inhibition prevent cytokine-mediated CXCR12 production in bone cancer cell line. First of all, CXCR12 is originated and identified as a homing chemokine in respect with white blood cells, and it is recently play important role in cancer progression due to their effect on the cells within the tumor microenvironment. And it is released from endothelial cells, fibroblasts, and immune cells. And it is effect through binding to desmin receptor CXCR4. And there are many drugs, people made these drugs that targeting CXCR4, and these drugs showed limited clinical success. Also, in this study, I want to focus on a specific approach that targeting the CXCR12 production. And there are many factors induce CXCR12 production, such as hypoxia, growth factors, and the one that I want to focus on is interleukin beta-1 beta, IL-1 beta. And the main signaling pathways that are regulated by IL-1 beta, as we can see in this slide, on the left-hand side, the canonical NF-kappa-B pathway, and this, the IL-1 beta activated the canonical NF-kappa-B pathway through the IKK complex, and also phosphorylation, the IKK complex by the alpha, beta, and gamma, and also activate the main component IKP alpha. Also, on the right-hand side, we can see the IL-1 beta also activated the MAP kinase pathway, which are JNK and B38 and ERK. These are the main components for MAP kinase pathway. So our aims in this study are assess the effect on CXCR12 production induced by IL-1 beta, and also block specific signaling pathways activated by IL-1 beta. And I used UTOS osteosarcoma cell line transfected with luciferase and linked it to CXCR12 promoter, and the CXCR12 induction done by using luciferase reporter assay, and the signaling pathways were assessed by using Western blotting using multiple phospho and total antibodies. At the beginning, my experiment starting with the identification, the time course, and concentration dependent for the IL-1 beta induced CXCR activity. And as we can see in this figure, the IL-1 beta stimulate CXCR12 production that's starting at 2 hours and reach a maximum at 6 hours, as in figure A. And on the right-hand side, the IL-1 also stimulate in a concentration between 3 to 10 nanogram per mole. After that, I starting the assessment, the signaling pathways on the CXCR12 by using the pharmacological inhibition, with, as we can see, for the MAP kinase inhibitors, by using 3 inhibitors, which are on the left-hand side JNK inhibitor, and in the middle ERK inhibitor, and on the right side B38 inhibitor. These 3 inhibitors in the luciferase reporter assay showed a minor effect on CXCR activity induced by IL-1 beta. However, these inhibitors significantly inhibit their main pathways, as for example, as we can see on the plot on the bottom, for JNK inhibitor, which is SP inhibitor, significantly inhibit JNK phosphorylation. And for other inhibitors, I need to test it in the future just to confirm it is involved in pathways and not involved in the reporter activity. Also, I tested 2 selective inhibitors for NF-kappa B pathways, which are SU1261, novel and selective IKK alpha inhibitor, as you can see on the left-hand side. And also, on the left-hand side, the IKK beta inhibitor, IKK2X1. These 2 inhibitors in reporter assay, the SU1261 on the left-hand side significantly inhibit the CXCL activity, while the IKK beta inhibitor doesn't affect the activity. However, these 2 inhibitors, as we can see on the plots on the bottom of the slides, for the IKK alpha inhibitor, significantly inhibit the P100 phosphorylation. I measured it because it is a non-target for IKK alpha, and also the IKK beta inhibitor significantly reverse IKB alpha and inhibit B65 phosphorylation, which are also main components for IKK beta. And also, I assessed the upstream kinase to see the effect on their signaling pathways. And as we can see, the TAC1 inhibitors I started assessed, which is 5Z7-oxozinol, abbreviated as 7-oxo. And this inhibitor on the signaling pathways, as we can see on the panel for figure A, the 5Z7-oxo significantly inhibits P100 phosphorylation in a low micromolar range, as you can see here, and also, and this is starting at the low micromolar range. Also, the inhibitor inhibit the components for IKK beta, P65 phosphorylation, and reverse IKB alpha. Also, for the MAP kinase pathway, the inhibitor inhibit JNK phosphorylation, and we know these 2 inhibitor pathways doesn't involve in the CXCR12 activity from the luciferase reporter. So, in conclude these, the inhibitors, that's IKK beta and the JNK MAP kinase, not involved in CXCR12 activity. Moving on the testing the 5Z7-oxo as in the reporter assay, the inhibitor inhibit the CXCL activity, as we can see in this figure, in mid to high concentration, and the effect starting at 10 micromolar, and about the IC50 was about 5 micromolar, and I was concerned due to this concentration. I was concerned about the cell viability, so I did the MTT assay, and I found, as you can see in this figure, the cell viability was about 70% at 10 micromolar. And because those 2 things, the concentration and the cell viability, I did the siRNA TAC1, and following 72 hours treatment, as we can see, the TAC1 run down very good and compared with the non-targeting control. And in the luciferase reporter assay, there is about 30% reduction in CXCL12 activity, and this is great. And when we moving to the signaling pathways, the siRNA TAC1 significantly inhibit JNK phosphorylation, as we can see here, and this indicates the effectiveness of the siRNA TAC1. But in the P100 phosphorylation, there's a mild effect about 30 to 35 reduction, and however, the siRNA TAC1 didn't affect the IKB alpha. And in conclusion, the pharmacological inhibition showed no role for MAP kinase and IKK beta, but the IKK alpha was involved. And the 7-oxo strongly inhibits CXCL12 production and all 3 main pathways. And the TAC1 siRNA showed a small inhibition to CXCL12 production, and this reflects the variation in the signaling. However, the TAC1 doesn't play any big role in CXCL production. suggesting 7-oxo may be off target. And lastly, I want to thank my group, Professor Robel, and Dr. Margaret, Dr. Katrin, and my best friend, Christy, for their support and help. Thank you for your attention. Sorry for the time.
Okay, we have time for a couple of questions. There are 4 mics down here, so if you have any questions, please approach the mic. Farhan, have you tested the compound against other cell lines?
For now, I just focused on the U2S cell line because, as you know, the expression of CXCR12 is also high on the osteosarcoma cell line. So after that, when I establish the effect on the U2S cell line, maybe also in our plan to test it in other cell line, such as breast cancer cell line.
Any question from the audience? Do you have plans to test the compound in any preclinical animal models as well in the future?
To be honest, we have not planning for the animals, not now. Yes.
Okay, if there's no question, please join me to thank Farhan again for the presentation.
Thank you.
The next speaker is Leevi Halonen from Tampere University.
Thank you for the introduction. Good afternoon, everyone. It is with great pleasure that I'm here to present my results here in the 90th World Congress of Clinical and Basic and Clinical Pharmacology. I am Leevi Halonen. I'm a physician and also a clinical lecturer and PhD student under Eva Moilanen in the Immunopharmacology Research Group, Tampere University. And it's my honor to present the results of our amazing team, myself, Professor Moilanen, and Doctors Pemmari, Nummenmaa, Hämäläinen, Moilanen, and Vuolteenaho. First, some background. We're studying transient receptor potential channels, which are a group of cation channels that are involved in various physiological processes, and particularly in pain and thermosensation. They have been emerging topic of pharmacology studies in a lot of different disciplines of medicine right now, and they were also awarded a Nobel Prize in 2021. Here you can see transient receptor potential anchorin 1 or TRPA1 that our group has extensively studied. Another subject of this study was osteoarthritis, which is historically known to be or historically understood to be a mechanical wear and tear disease, but nowadays we know that it is a chronic inflammatory disease that is affecting the whole joint. It is a leading cause of disability among aging population, and there is still no effective disease-modifying drugs discovered. So we don't really have any medication to properly slow down the progression of osteoarthritis or to reverse it. The pharmacological management of osteoarthritis focuses mainly on the pain relief. And our group has earlier discovered that the TRPA1 channel is functionally expressed in human OA chondrocytes, where it is involved in upregulation of inflammatory factors such as interleukin-6. And this served as our inspiration to investigate the expression of other possible TRP channels that might be present in primary human chondrocytes. And how we did it is first we ran an RNA-seq analysis on OA chondrocytes and obtained all the TRP channels that we were found to be expressed. Next, we wanted to confirm the results of RNA-seq on the most expressed TRP channels. Third experiment, we want to assess the effects of ibuprofen and dexamethasone, both drugs commonly used to manage OA pain, on the expression of aforementioned channels. And lastly, since, spoiler alert, TRPM8 was found to be expressed in these chondrocytes, we want to see whether TRPM8 and also TRPA1 agonist menthol might have any effect on these chondrocytes. Okay, some methodology. We get the cartilage samples from knee replacement surgery, so from real patients. We culture them with different combination of stimulants, run the RNA-seq on the chondrocytes, and then we also run a PCR on the other experiments. So on to the interesting part. Here is our results of our RNA-seq. On the left-hand side, so you can see the table here. We had a control chondrocytes, and then we also stimulated them with interleukin-1 beta to see whether inflammatory stimuli affects the affects the expression of that TRP channel. And you can see the fold change is mentioned when it was found to be statistically significant. On the right-hand side, it's the same data, but it's presented as heat map. And I want to point out a couple highlights here. Firstly, the TRPA1. Down here, you can see the pretty massive upregulation when stimulated with interleukin-1 beta, which was also shown earlier by Numema and company. We here also have TRPM7 really highly expressed, and also V4, C1, and M8 with slightly smaller counts, but still quite significant counts. And we decided to choose the 4 most expressed TRP channels here for further experiments. And also we want to choose TRPA1 for kind of positive control, and also because it is so reactive to inflammatory stimuli. And now after we chose our 5 channels, we actually run the PCR on the same channels. This is done on a different set of patients. Those are patients in the RNA-seq and on this PCR are completely different, but the results are still pretty much the same. We can see the massive, tremendous upregulation of TRPA1 when chondrocytes are exposed to interleukin-1 beta. We can see the downregulation of TRPC1, and also slightly bigger downregulation of TRPM8 here, while TRPM7 and TRPV4 were not affected by interleukin-1 stimulation at all, which, as we can see here, are in concordance with our RNA-seq results. Then we wanted to test the effects of dexamethasone and ibuprofen. Both are commonly used medication in OA pain. And we actually found that the dexamethasone seems to attenuate the effects of interleukin-1 beta on TRPA1 expression, and also slightly less on TRPM8 expression right here. But dexamethasone had no effect on the expression on TRPM7, TRPV4, or TRPC1. We also tested ibuprofen, but it had no effect on the expression of these aforementioned channels. Next, we want to test the effects of menthol, which is kind of interesting compound. It is a commonly used cooling agent used in many ice gels, and is naturally found in, for example, peppermint and other mints. It is traditionally known to be an agonist for TRPM8, and the cooling sensation of, for example, menthol powders, menthol gels are thought to come through TRPM8. But there's also studies showing that menthol also has some bimodal function on TRPA1, with micromolar concentration activating TRPA1 and higher millimolar concentrations actually inhibiting TRPA1. But these concentrations we used here are on the activation area of TRPA1. And lo and behold, menthol actually seems to dose-dependently slightly upregulate the expression of cartilage-degrading matrix metalloproteinases 1, 3, and 13. And it also, at 60 micromolar concentration, seemed to upregulate the expression of inducible nitric oxide synthase. And also a trend was seen to increase interleukin-16, but there is no statistical significance there. So to sum up our results, old news is that we have shown that TRPA1 is expressed in OA chondrocytes, but now we have also showed the expression of 19 other TRP channels in chondrocytes, with TRPM7, V4, C1, and M8 having the highest expression levels. To my knowledge, the expression of TRPA1 in primary human chondrocytes has not been described in literature before, and TRPA1 was found to be most responsive to inflammatory stimulation. The TRPM8 and TRPA1 agonist menthol increases the expression, seems to increase the expression of MMP1, MMP3, and MMP13, and also INOS and probably interleukin-6. And well, these findings support the role of TRPA1 in osteoarthritis, but it also displays TRPM8 as a potential novel drug candidate for treatment of OA. And we have also now described all the TRP channels found in osteoarthritic chondrocytes for further studies. And lastly, I want to thank the Immunopharmacology research group and all my colleagues there. Check out our other presentations, as can be seen on the right-hand side, with Dr. Vuolteenaho presenting her results very soon. And also, if you want to see these published results and more methodology, you can see our published article through this QR code. Thank you for your time.
We have time for a couple of questions.
Thank you very much for your lecture, very interesting. I wonder if you have the chance to check TRPA1 and the other channels in diseases also related to the joints, such as rheumatoid arthritis, psoriatic arthritis, and you have seen the same pattern, yes or no? Because the same drugs also are given in these rheumatic diseases.
Yes, they are most likely also involved in other arthritis types too. I have seen like some urine models that test effects of menthol and TRPM8 agonism on mice, also where. So yes, it's the answer.
Yes. Okay. Thank you.
Hi, enjoyed your talk. Could you give me some indication about the promoter regions on the different TRP channels? Because obviously interleukin caused a huge increase in TRP1, but it showed negativity on the others. So is there any correlation with respect to the binding sites, signaling binding sites on the promoters?
That is actually a really great question. Thank you for the question. Unfortunately, I have to quite confess, I have not like checked out the promoter sequences on TRPA1 or M8, so I can't answer that one. But I'll be sure to check that out right after this lecture.
To confirm the function of TRPA1, have you done any gene editing experiments, like knock-in or knockout experiments to correlate with the phenotype?
For TRPA1, you mean?
TRPA1.
Yeah, we have like Numben Ma and like others have published results also on knockout mice, and also they have used TRPA1 antagonists to confirm that the rise in interleukin-6 expression is through TRPA1 at least.
Okay, if there's no more question, please join me to thank Leevi one more time for his excellent presentation. Okay, our next speaker is Katrin Vuolteenaho from Tampere University. Please come on stage.
Yes, thank you. And thank you for the opportunity to present our results on adipokines. And I am Katrin Vuolteenaho. I'm clinical lecturer from the Immunopharmacology Research Group, Tampere University, so same place as Leevi previously. And in this talk, I present results from adipokines in osteoarthritis, and especially the recent results about adipokine Meteorin-like protein as a novel factor and potential drug target in osteoarthritis. But first, a bit about background about osteoarthritis and adipokines, why to study them. These are facts from the osteoarthritis white paper that was published by ORC a few years ago, showing the importance of this disease. Osteoarthritis affects 1 in 4 adults age 40 and over. By 2032, OA will affect 1 in 3 adults as population ages and obesity rate rises. And 50% of all people with OA are of working age, so it's not just a disease of the old. It causes pain, it causes impaired function and reduced quality of life. And the drug treatment is symptomatic. It can reduce pain, but there are no approved drugs that could prevent. There are no good drugs to slow down the progression of osteoarthritis, so really worth investigating. So age obesity increased the incidence of osteoarthritis. This is a study by Reyes et al., which was done in a population of 1.7 million people who were over 24, who had no osteoarthritis, and they were followed for 4 years. And the result was that yes, obesity does increase the risk of DOA and hip OA, and yes, this is not surprising. But obesity also increases the risk of hand OA. And that tells us that obesity is not just increased load on joints. There must be some systemic factor that links obesity to osteoarthritis unless you walk with your hands a lot. And that is where adipokines come into play. Adipokines are mediators that were originally found to be produced by adipose tissue, and they are related to regulation of appetite, regulation of energy expenditure, and so on. But nowadays we do know that they are produced by many other cell types. They have receptors in many cells, and they have been linked to low-grade inflammation, metabolic syndrome and related diseases, and lately also to osteoarthritis. And we and others have studied this subject quite extensively, and in OA patients, adipokines such as leptin, adiponectin resistin, and retinol-binding protein 4, they associate with measures of inflammation and cartilage breakdown, like MMP enzyme, like IL-6, like nitric oxide, in synovial fluid of OA patients, and in cartilage medium. They also associate with measures of OA severity, measured by knee society score, radiographs, or biomarkers of of severity of osteoarthritis like MMP3 and COMP. And further, we have also shown that chondrocytes respond to leptin and adiponectin producing enzymes and pro-inflammatory factors that are related to osteoarthritis. So, now we know that there are adipokines in synovial fluid. We know that they are related to the pathogenesis of OA, but where do they end up into the synovial fluid? Well, of course, adipose tissue produces adipokines, and they are transmitted to circulation, and they are diffused in the synovial fluid. That's another one possibility. And in addition to that, also different joint structures also have been shown to produce adipokines, and also the fat pads that are in the vicinity, near vicinity of the joint structures, like infrapatellar fat pads. And so they are really in the close vicinity of cartilage, which is the main focus of osteoarthritis pathogenesis. But in this present study, we hypothesized that also chondrocytes, the only cell type in the cartilage, and they are also mesenchymal cells, just like adipokines. They might also produce adipokines that could be involved in osteoarthritis, and this was our hypothesis. And we started to study this. We recruited 10 osteoarthritis patients that were undergoing knee replacement surgery. We had cartilage samples from them, which are waste material from this kind of operations. We did cell culture experiment, and then we extracted the RNA and did RNA sequencing to study all messenger RNAs that are expressed in these cells. And this data was published by Antti Pemmari. And in the present study, we analyzed the same data and focused on adipokines. And what we found was that the most expressed adipokines within the chondrocytes were bisphatin and Meteorin-like protein. They stand out from the data. And further, when we incubate these cells with an interleukin-1, their expression is increased. We did want to confirm these results, and we recruited 10 more OA patients, did the same kind of thing, extracted the cells, but now we used quantitative RT-PCR to measure just this bisphatin and Meteorin-like protein. And we were able to show that yes, and confirm the results that yes, they are expressed in OA chondrocytes, yes, interleukin-1 induces their expression. And further, in these experiments, we treated the cells with glucocorticoid dexamethasone and NSAID ibuprofen, both of drugs that are used in the treatment of osteoarthritis. And we showed that dexamethasone increased further IL-1 induced bisphatin expression and downregulated Meteorin-like messenger RNA expression, while ibuprofen decreased somewhat bisphatin messenger RNA expression, but did nothing to the Meteorin-like messenger RNA expression. So they do respond to the drugs used in the treatment of osteoarthritis. When then wanted to analyze in our osteoarthritis sample collection of 100 OA patients and wanted to analyze whether these adipokines, visfatin, Meteorin-like protein, do and are reflected in these patient samples. So we have from these patients plasma samples, corresponding synovial fluid samples, and corresponding cartilage culture medium samples. And we measured from these samples visfatin and Meteorin-like protein levels, and we really did find visfatin in plasma and synovial fluid samples, and we did find Meteorin-like protein in plasma and synovial fluid samples, and also in cartilage culture medium samples of these patients, all in nanograms per millimeter level. And then a few highlights about these results. When we compare the levels and concentrations of Meteorin-like protein, we found out that in these patients, Meteorin-like protein levels were considerably higher in the synovial fluid samples than in the corresponding plasma samples. We also saw a nice correlation between these two. So concentrations in synovial fluid samples correlated with those in plasma samples. We also saw a correlation of synovial fluid sample Meteorin-like protein with the cartilage culture samples of Meteorin-like protein. And these results point to in that direction that really cartilage might really be a significant producer of Meteorin-like protein. We then also analyzed whether these adipokines do correlate with MMP enzymes and IL-6, which are both related to the pathogenesis of osteoarthritis. And we could show that Meteorin-like protein is correlated with MMP enzymes, with IL-6, within synovial fluid and within cartilage culture media, while visfatin showed no such correlations. So in conclusion, as we hypothesized that good chondrocytes as mesenchymal originated cells, kind of like adipokines within the cartilage, could produce adipokines. So we, in this study, could show that really they are highly expressed adipokines in genome-wide expression analysis in these primary human OA chondrocytes. And we also show that these chondrocytes do respond to IL-1 and glucocorticoid producing Meteorin-like protein when IL-1 induced and downregulated by glucocorticoid dexamethasone. We also showed that Meteorin-like protein is secreted from the cartilage into the synovial fluid in significant amounts within the OA joints, and we also showed that these are associated with these cartilage-degrading MMP enzymes and pro-inflammatory IL-6 in OA patients. So these preliminary findings, they really do give us idea that this novel adipokine could be involved in the pathogenesis of osteoarthritis and could be a potential drug target for disease-modifying drug development. So, last, I would like to, of course, thank all the collaborators and authors of this abstract. We had Anna Koskinen-Kolasa, who has done really a great amount of work with adipokines and studied adipokines in osteoarthritis and defended her thesis about it. We have Antti Pemmari, who has been the bioinformatic guy analyzing these next generation sequencing results. We have Mari Halemäläinen, who have done and supervised this lab work that was done, and Eeva Moilanen for supervising our research group. And of course, important thing is this Coxa Hospital for joint replacement, and Teemu Moilanen, who recruited these patients, and that's how we obtained these cartilage samples. And yes, thank you, our research group. And as Leevi mentioned, we have a few great presentations coming up in the following days, so you are most welcome to listen to them also. Yes, thank you.
We have time for 1 or 2 questions. I noticed that the BMI of the 100 patient is relatively high.
Yeah.
I'm just curious if the Meteorin-like protein, is it also produced by the adipocytes?
Yes, it is produced by adipocytes, but when we have studied these adipokines in osteoarthritis patients, so the most significant correlation with BMI is with leptin. So leptin is the most correlated to BMI, but in fact Meteorin-like protein is not correlated. We did check that.
Okay. Also, just out of curiosity, do these patients also suffer from the hand OA besides the knee osteoarthritis?
Well, of course, knee osteoarthritis is the primary concern because they are heading to the joint replacement surgery, and yes, it would be of interest to have also samples from these hand OA patients, but that's not so simple as from the knee replacement surgery. So we unfortunately not have those samples.
Okay, if there is no further questions, please join me again to thank Katrina for her presentation. The second speaker, Lizanne Silva, she has not submitted her slide deck, so we will not be holding that talk. So this will be the end of this oral communication session. Please check your schedule for the next talk. Thank you for your participation.